There we go. Okay. There's our missing folk. You guys had an AMP lab test, I think. How'd that go?
Wednesday we had a lab practical and today we had like so much.
The lab practical go. Not bad. Okay. All right, well, here. Well, I started in with
Find one.
That's pretty loud. Testing. Test. Yes, that works. Okay. There we go. Hey, the crew's taking a quiz right now. That's why we're quiet.
Okay.
I will email that quiz to you, in fact. I need to do that. So
Okay,
later
perfect.
today. All right.
Okay, sounds good. Thanks.
How you feeling?
I'm feeling good. My plan is to come back next week in
Oh.
class. Yeah.
That's fast.
Yeah. I hate this online Zooming, trying
Yeah.
to figure everything out by
Yeah.
myself. So I figured it'd be best for me, like in my classes, to be back.
Okay. All right. We'll make a space for you and your crutches.
So.
Okay. Let's get with it. Okay. Very strange. All right. Go on you. There it goes. All right. Now, stopped last time talking about these major histocompatibility complex proteins. So this is a big gene complex on chromosome 6. And this big gene complex encodes proteins that are very important for the immune system's determining of what is you and what's not you. And there are two main types of these proteins. There are class 1 proteins and class 2 proteins. Since these proteins are called human leukocyte antigens, they're given the abbreviation HLA. So HLA refers to the protein products. MHC refers to the genes that encode protein products. Class 1 proteins are found on the surfaces of every nucleated cell in the body. Every nucleated cell in the body has a class 1 MHC protein. Only antigen-presenting cells have class 2 HLA proteins. So antigen-presenting cells would include dendritic cells, macrophages, B cells. That's about it. So if you are a professional antigen-presenting cell, like a dendritic cell, macrophage, or a B cell, you have class 2. So class 2 MHCs are restricted to certain cell types. Class 1s, they're found on every nucleated cell. So, will red blood cells have type 1, have class 1 MHCs? No, they won't. They will not. Very good. So how do these things go about being the barcodes for your cells? Remember that the genes that encode these proteins are extremely polymorphic. In other words, they're incredibly variable from one person to another. That's the reason why these barcodes are so different on your cell to the other cell. We use the example of Liesl donating skin to Zoe after her burn. That's very kind of Liesl, but it wouldn't help Zoe because her immune system would see those class 1 proteins on the surfaces of the skin cells, and the immune system would recognize them as foreign. It would attack them and it would reject them. And then if there are any immune cells in Liesl's skin, those cells might come out and actually attack Zoe's tissues as well. That's called graft versus host response. The graft versus host response. This is a problem in transplants, particularly transplants that don't need to be matched all that well. Livers, for example, you need the right blood type. That's pretty much it. Kidneys, on the other hand, you got to match those a lot better. They're not going to last nearly as long. So this particular slide is a figure taken from your book. There are 3 different genes in the class 1 MHC gene cluster, and they are called HLA, A, B, and C. And then there are 3 main class 2 MHC molecules, and they're just called DR, DP, and DQ. The class 1 MHC molecules, when they are made, remember, these are membrane proteins, so they begin their life on a ribosome that gets taken to the endoplasmic reticulum. So they're translated at the endoplasmic reticulum, and if there are any endogenous foreign substances, those will bind to the little cleft of the class 1 molecule, and they will then be trafficked to the surface. And so that endogenous antigen is presented on the surface of the cell. I actually have some pictures of these molecules that I put on our Blackboard site. Where's our, oh boy, let's see here. There we go. All right, so let's get this so that Miss Emma can see these as well. So here's a class 1 MHC. You can see it has 3 different parts of the polypeptide. This is all one protein here. So this alpha 3, alpha 2, alpha 1, that's one protein. This piece down here, this beta 2, that's called microglobulin. Microglobulin, unlike the MHC class 1, doesn't vary from person to person. So it's a really important structural piece. But this little cleft right there, when this gets put into the endoplasmic reticulum, any viral proteins being made inside the cell, they actually get pumped into the endoplasmic reticulum. And these antigens bind there. And then this gets presented to the surface of the cell. Now, the class 2s, remember, with class 2s, you have cells that phagocytize foreign material. That foreign material goes to a phagosome, which fuses with a lysosome. And these class 2 proteins are in the lysosome. So they will bind anything from the lysosome. And then this gets separated from the lysosome and then gets trafficked to the cell membrane. So the difference between these two when they're at the cell membrane is this. Let's go back to our slides. These class 1 molecules that are presenting their antigen, they will be recognized by T cytotoxic cells. And T cytotoxic cells, remember, express a surface protein called CD8. And if the T cell receptor of that T cytotoxic cell recognize it fits with that antigen presented on the surface by the class 1, the T cytotoxic cell now has license. It's given the go-ahead to destroy that cell, because that tells the T cytotoxic cell that this cell is infected with viruses. Or maybe it's infected with an intracellular parasite or something like that. And the T cytotoxic cell destroys that infected cell. Now think about this with me. Do you guys know anything about the disease malaria? You get malaria from a mosquito. The mosquito injects sporozoites. Where do those sporozoites go? Where's the first place they go? It's right here. You meet the diaphragm. It's big, it's red, it's fleshy. You look at it under the microscope, looks like the laxodons, looks like apentiles. Starts with an L, ends with an R. Liver, thank you. Goes to your liver. And it divides in the liver, and then the merozoites come out and they infect red blood cells. Get lots of red blood cells that are infected. And those red blood cells present those antigens to these cytotoxic cells. No, they can't, because they don't have class 1 MHCs. Even though red cells are short-lived, relatively speaking, they escape the immune response by infecting red blood cells. Because red blood cells can't tell the T cytotoxic cell unaffected. They don't have the class 1s. So that's how malaria essentially escapes your immune response, because it infects a cell that can't present the antigen. Now, what about these other antigen-presenting cells? Macrophages, dendritic cells, B cells. They can take that MHC2 bound antigen presented on the surface, and that will be recognized by a T helper cell. And these cells express a surface protein called CD4. And if the T cell receptor on the surface of that T helper cell recognizes that antigen, if it fits the T cell receptor, the antigen fits the T cell receptor, the CD4 molecule is able to also make an association with the MHC class 2 that activates the T cell. And now the T cell will expand, it will undergo clonal expansion, and it will beef up macrophages and B cells that will make antibodies against that particular antigen. So the B cell essentially licenses the T helper cell helps the B cells beef up their antibody production. It helps the macrophages fight the infector. So here's a little cartoon to try and give you an idea of how the cow eats the cabbage. when it comes to antigen presentation. So here's your class 1. Class 1s are expressed in the ER. So you probably learned from A&P, the early days of A&P, about protein synthesis, that they occur on ribosomes, but proteins that are embedded in membranes or end up in vesicles to be secreted, that translation is stopped, and then the ribosome is dragged over and attached to the ER. And then this protein is made inside the ER. And then these antigens are imported inside the ER, and they bind, as you can see here, the MHC class 1. So MHC class 1 proteins present antigens that occur inside cells. It's an important distinction to understand, inside cells. These vesicles then get pinched off, and they go to the cell membrane, and there you are. Presents that antigen. And what recognizes this antigen? CD8 expressing T cytotoxic cells. Whereas your class 2 MHCs, they're expressed in the phagolysosome. So here is your phagolysosome there, and here are your class 2s. They take up proteins and other antigens that are found in the lysosome. How did they get to the lysosome? They came from the outside. So the class 2 MHCs will present antigen that came from the outside. Then once these bind the protein, these are trafficked. They're sorted essentially to another vesicle that goes to the membrane. And these are recognized by CD4 positive T helper cells. And that's how CD4 T helper cells can get activated. Now, Aaron, you asked a really good question on Wednesday, and I hope this will partially answer your question. How do then we get autoimmune diseases? Turns out that particular alleles of these class 1 MHCs do a better job of grabbing self-proteins and presenting them on the surface than the others. That's why certain genotypes of these MHC genes are associated very strongly with certain autoimmune diseases. And it simply comes from the fact that these self-proteins get imported into the ER, they're able to bind and then get presented to the surface, and the T cytotoxic cells will recognize them in some cases. It takes a while to get a T cytotoxic cell with a T cell receptor that can recognize it. That's why these diseases don't usually manifest very early in life. They take a little longer. And also, many autoimmune diseases are more common in women than in men, because it turns out female sex hormones influence the tendency for the immune system to present its own antigens. Does that maybe partially answer your question? It's, of course, more complicated than that, but I gave you the Reader's Digest version, and that will have to do for now. So these collaborations between these cells and the recognition of the TCR means T cell receptor, that's the antigen receptor on the surfaces of T cells. When you have a complex between the HLA protein and the antigen, so if this is, say, a class 1 protein and the T cell receptor and the CD8 protein, that complex activates the cell. And likewise, when you get this complex between the HLA class 2 and the antigen. And the T cell receptor and the CD4 protein. That will activate the T helper cell. And T cells, when they get activated, one of the first things they express is interleukin 2. Interleukin 2. And interleukin 2 is an autocrine. An autocrine activator of T cells. Autocrine means it makes its own activator, which is interleukin 2. Same thing here. These make interleukin 2. And in response to interleukin 2, they divide, they expand, they undergo clonal expansion. So as I said, the helper cells, they will help B cells mature, make antibodies, so on and so forth. They'll facilitate, they'll magnify the interaction between the antigen presenting cell. And remember what the antigen presenting cells are. They're usually dendritic cells. So I'll say DC, dendritic cells, or macrophages. And the lymphocytes, the B cells whose surface antibodies recognize that antigen, they are activated by the T cells. There are two main types of T helper cells, T helper 1 and T helper 2s. T helper 1s provide humoral immunity. When you see humoral, that doesn't mean it's funny. It comes from the old term humors, meaning things found in secretions, things found in liquids like blood or mucus or something like that. So it refers to the production of antibodies usually. And the T helper 1, T helper cells, they develop cell-mediated immunity, meaning they beef up the activity of neutrophils and macrophages and what have you. And they do this by expressing different types of cytokines. Now, we will see a couple of examples of particular antigens that don't need antigen-presenting cells to activate the T cell. These particular antigens can bind to unactivated T cells. When they bind to those unactivated T cells, they activate the T cells illicitly. And when they activate the T cells illicitly, they really activate them. And these are called superantigens. And the best example of these superantigens is the toxic shock toxin made by certain types of Staphylococcus bacteria. Toxic shock essentially results from hyperactivation of the immune system. And you get this thing in your body called a cytokine storm. You get very leaky blood vessels, you get a very high temperature, and it's a medical emergency. And that's due to these toxins made by these strains of Streptococcus and Staphylococcus bacteria that can just bind T helper cells and activate them illicitly. So they cause fever, they cause shock, and it's not fun for the patient. So clonal selection, we talked about this last time again. It's due to the fact that a B cell is activated if and only if its antibody binds to that antigen relatively tightly. And then as the B cell expands, remember, as it replicates its antibody genes, it uses a DNA polymerase that makes mistakes. So as it expands, you get some variability in the types of antibodies they make. So which of those subtypes of the expansion get activated? The one whose antibodies fit the antigen the best. And that's called clonal selection. That's the reason why antibodies made later in the immune response fit the antigen better than the ones made earlier. And due to cytokines made by T helper cells, they will eventually switch the subclass of antibody that they make. Remember, the first antibody they secrete is Ig what? M. IgM. M for mature. I'm mature now. I secrete antibodies. So IgM. And then it undergoes the class switching, and that is the result of cytokines made by T helper cells. Which antibody subtype will it make? Again, depends on the cytokines made by the T helper cell. So the T helper cell has the antigen presented to it by the B cell. The T helper cell gets activated by this interaction between the MHC class II, right there, and the CD4 protein right here, and the T cell receptor that bears the antigen, all right there. That complex sends a signal to the inside of the B cell. Sorry, to the T cell. The T cell then, in response to that activation, makes cytokines. One of them that it makes is interleukin-4. Interleukin-4, and then some others as well. And those others as well essentially tell what kind of isotype switch this antibody, this B cell is going to do. Will it make IgA? Will it make IgG? Will it make IgE? Depends on the cocktail of cytokines it gets from that T helper cell. Now, there is another group of T cells that are called T regulatory cells. T regulatory cells turn down the immune response. So once T cells are activated, the T helper cells will expand, and then later on, the T regulatory cells will start to expand as well. And as these cells start to run out of antigen, those T helper cells are going to come and shut them down. So these T helper cells, they secrete interleukin-10. Interleukin-10 tends to shut down the immune response. And then the other cytokine they will secrete that will tend to downregulate the immune response is TGF-beta. Transforming growth factor beta. My F looks a little there. T regulatory cells are also activated by very small amounts of antigen. If you're constantly exposed to really small amounts of antigen, The T regulatory cells are activated without the helpers being activated. So that'll actually prevent you from responding to the antigen. And that's why things that you grow up with, you don't tend to react to them. That's why if you look at research on kids who grow up in rural areas, farms, a lot of work's been done with the Amish, for example. So the Amish raise their own farm animals. They do their own farming, they raise their own food, so on and so forth. Amish kids have among the lowest levels of asthma and allergies and things like that, because they're constantly exposed to low levels of these particular antigens. So the regulatory response is very high in these kids, and that prevents them from getting these autoimmune diseases. How many of you have heard of allergy shots for kids with allergies? Those contain really small amounts of very highly purified, very solubilized antigen. You give a shot intramuscularly, this stuff then will start to leak out into muscle very, very slowly. It's too little to activate the T helper response, but it activates that T regulatory response. And as you get those shots, eventually the T regulatory response will start to take over and the allergies will start to go away. That's a strategy behind these allergy shots. And then after an immune response, you always get a population of cells that are left over, and those are your memory cells. Memory T cells can last for decades. Decades. And this is mainly the reason why your COVID-19 shot, which really doesn't protect very well against the new variants, the Omicron-based variants, and so on and so forth. But you still have memory T cells from that initial COVID-19 vaccine you may or may not have had. And that's why the shots will tend to protect against hospitalization and severe disease, because there's still a base antigen response that you have that pretty much all these SARS-CoV-2 viruses have. And those give you enough protection to prevent the virus from causing the acute respiratory distress syndrome that the virus can cause. So as we said, the T cytotoxic cells, when they are activated by that complex, they will secrete this cocktail of nasty things. So perforin makes a little pore in the membrane. So if this is the virus-infected cell, the perforin inserts itself into the membrane of this cell. And then the granzymes are these enzymes that then go through the pore, get into the cell, and these chop up things inside the cell and cause the cell to off itself. So these induce programmed cell death, otherwise known as apoptosis. And this cell gets the deep six. So if the cell is virally infected, it's an abortive infection. If it has an intracellular parasite, that's an abortive infection. So the cell gives its life so your body may live. Moment of silence. Oh, that's enough. Natural killer cells. Natural killer cells are interesting cells. They have receptors on their surface for damaged proteins. So if it comes to a cell and there's enough damaged proteins on the surface, the natural killer cell will bind to those damaged proteins and will secrete a cocktail of entities that kill the cells. The other thing these cells look for is if the cell has antibodies attached to it, it recognizes those cells as foreign, and it uses those antibodies to kill the cell. And then if the cell expresses a HLA class 1 protein that doesn't look right, it'll recognize that and it'll kill the cells too, because often cells that express whacked out HLA-1 proteins are cancer cells. And so they're very important also in killing virally infected cells, cancer cells, and damaged cells. And we talked about these cells already, so I'm not going to talk more about those. This just shows you the killing mechanism. Is a really nice picture showing a tumor cell right here with the T cells attached to it. There's a very exciting therapy for aggressive cancers today known as immunotherapy. Turns out the immunotherapy, cancer cells secrete molecules and also put molecules on their surfaces that take these lymphocytes and activate them. T cells come in saying, I'm going to kill you, I'm going to kill you, I'm going to kill you. And the cancer cell secretes these molecules or has molecules on their surface, and the T cell gets there and says, why was I here? Okay, I'm going to go wander off this way and just, I don't know, take up knitting or something like that. So this new therapy called immunotherapy actually gets these T cells to remember why they were there. And now these T cells hone in on this tumor and kill it dead. It's called immunotherapy. Immunotherapy is proving remarkably successful against treating very aggressive types of melanomas and ovarian and lung cancers. Yeah, we've talked about that. So how is it that the mother's body does not recognize the fetus as foreign? So the fetus, it turns out, can make IgM molecules, but it can't make these other more mature antibodies. And the fetus has the placenta, and the placenta turns out to be a very important immunosuppressive organ. So maternal immune cells come to the placenta, they see something foreign, but they say, no, you live for now. And they keep doing that. They keep going to the placenta. The placenta has these cell surface proteins that just tell the immune system, back off, and it does. And then the maternal antibodies that the baby receives across the placenta, those are mainly IgG antibodies. Those will give you protection for about the first 6 months of life. And if the baby is breastfed, the baby will receive immunoglobulin A through the breast milk. And typically a baby is weaned 8, 10, 12, 13, 14 months, something like that. It varies pretty wildly. Moms who go back to work will usually express, they usually use a breast pump, and the baby gets immunoglobulin A that way. Usually by the time the baby, she starts solid foods, usually 4 or 6 months, somewhere around there. Usually the baby now is eating enough solid food to where you can wean the baby. By that time, the baby's immune system is now making its own antibodies, typically. Okay, go ahead, ask me. I know you're ready to ask it. Come on, ask it. What if a baby's formula fed? You want to ask that, right? Oh, you're not. Okay, I won't answer it then. The baby's formula fed, typically, formula fed babies will get more sicknesses than breastfed babies. That's a fact. But usually the kid is healthy enough to where it can fight off those infections. And then the immune system is subject to aging, just like anything else. So you get decreased T cell activity, your thymus shrivels, your thymic hormone production, that goes down as you age. Your antibody production, that goes down as you age. So consequently, as you age, you get sick more often. And your ability to handle sickness when you are older is not as good. Vaccines also, you don't respond to vaccines as well. That's why the flu vaccine for people over 65 is a genetically engineered vaccine that's really high concentration of antigens. Whereas the flu vaccine you might have gotten at Holton Health or at WeatherWax or wherever you got it is an attenuated flu virus. You get injected intramuscularly, it infects some of the cells, your immune response responds to it, and you have immunity to the flu strains. That's not good enough for people over 65. The immune response is poor. So you have to have the genetically engineered vaccine that's much higher dose in order for it to work. Consequently, many of you here will work with the aged. When you work with the aged, if you're sick, you really can't go into work. You have to protect those patients. And yes, you can wear an N95 or something like that, but very often seeing faces is very important for patient care because the relationship you have with the patient ends up being very important. If you're hiding behind a mask, which sometimes you have to do, if you're dealing with, patients who have cancer or something like that, you may have to wear a mask. But if you're doing home care or something like that, you don't want to go there in a mask, and you just need to not go if you're sick. All right, any questions about the immune system before we charge onward to something else? Reagan.
Does that mean to just finish the next chapter lead up to quizzes then? Or
No, I'll squish them all together. One quiz. I'm assuming you don't want two quizzes. Right. So put them all together into one. Okay, there we are. Very good. So I will get as far as I can on this. And the back end of this is actually podcasted and it's on the website. So I will let you guys get the rest of that. So cancer is derived from this Greek word for crab, carcinoma. Carcinoma sounds a lot like carcinoma, which is cancer of epithelial tissue. This is simply a disease that involves cells that divide uncontrollably. That uncontrollable growth then becomes a new set of cells that can then invade other tissues. So there is a term I need to introduce you to, that is hyperplasia. Hyperplasia means cells that overproliferate. So you have too many cells in that tissue or organ or something like that. So benign prostatic hyperplasia occurs in older men in response to 5-alpha dihydrotestosterone. The prostate gets bigger and it compresses the intermediate urethra. So men have trouble urinating. Is that prostate cancer? No, that's benign prostatic hyperplasia. The prostatic cells have divided. There's too many cells there, causing the symptoms that afflict older men typically. when we say cancer, usually we're referring to something that is damaging other tissues usually. So the term neoplasm a neoplasm refers to an abnormal mass of cells, the growth of which exceeds and is uncoordinated with that of normal tissues. So this abnormal mass of cells in a tissue, the growth of which exceeds and is not coordinated with normal tissues. So neoplasm simply means new growth. So if a neoplasm is benign, typically that means it grows relatively slowly. Also, when you take a biopsy of a benign tumor, it has a capsule around it, a fibrous capsule. The cells have not broken out of that capsule, so the tumor is not invasive. Now, benign tumors can impinge on nerves and blood vessels and other tissues and cause problems. But they're not invading other tissues. They're not actively invading other tissues. The cells of the benign tumor are relatively well differentiated. They don't look de-differentiated. They don't look immature. If you look for mitotic cells, you can get a percentage of cells that are dividing. That gives you what's called a mitotic index. The mitotic index of a benign tumor tends to be low. And they don't metastasize, meaning they don't spread to other organs. Malignant neoplasms, they grow rapidly. They are not encapsulated. They are invasive. They're poorly differentiated. They have a high mitotic index, and they can spread to distant sites. They can undergo metastasis. So that's the difference between benign and malignant neoplasms. So, for example, a fibroma. A fibroma is a benign growth in fibrous tissue. You can tell it's fibrous tissue, it's benign. Also a chondroma. Chondroma is a benign tissue of cartilage. And these benign tissues are classified according to their origin, tissue of origin, and their microscopic pattern and their, what have you, architecture. So here are some microscopic samples of benign and malignant tumors. So if we go up here under low power, can you see in the middle there, the top there, you see that space around the tumor? That's a fibrous capsule. That's encapsulated. Also, as you look at those cells, you can kind of tell what they are. So these cells over here, they look a lot like ductal cells, don't they? So those are well differentiated. They're not growing that fast. As you start to increase the power, you can even see the encapsulation here. And these cells, the mitotic indexes are not that high. There's a malignant sample there. See how integrated that is into the surrounding tissue? It's not encapsulated, is it? As you go higher, now your mitotic index is quite high. You don't have encapsulation, and these cells, they just kind of look like dots. They're dedifferentiated, they're not well differentiated. And there are various stains you can use to determine certain types of tumors and things like that. So this is what pathologists are looking for when they get tumors and they're staining them and looking at them. So your benign tumor typically has oma at the end of it. If you see oma, lipoma, leiomyoma, meningioma, they're benign. They may progress to full-blown cancer. Malignant tumors are named according to the tissues from which they arose. So carcinoma, if they arose from an epithelial tissue, they are a carcinoma. Most cancers are carcinomas. So adenocarcinoma, that's a tumor that arose from an epithelium found in ductal tissue, adenocarcinoma. Sarcoma. Sarcomas arise from mesenchymal tissue. Sarcomas. Do you know the difference between an epithelium and a mesenchyma? No. Think of a marching band. So marching band, everybody's in nice straight lines. And epithelial cells, they line up one next to the other. They have good solid junctions. They're nice and ordered. And then instead of a marching band, think of Woodstock. People just kind of milling around aimlessly, lots of space in between them. Oh yeah, we're over here, you're over there. I'm okay, you're okay. Yeah, just hang loose, you know. That's a mesenchyme. Lots of loose connective tissue. Cells are just kind of here, there, everywhere. So epithelium of your skin, that's an epithelium. The dermis is a mesenchyme. Make sense? You guys look at slides of skin and dermis, right? Yes. Looks like you did. Well, if you didn't, you have the slides and one of you can look at them and see the difference. So if a tumor arises from a mesenchyme, it is a sarcoma. A sarcoma. Blood-based tumors. If the tumor arises from cells of the lymphoid lineage, they are lymphomas. And if they arise from myeloid-based cells, they are leukemias. Leukemias. So here's an example of the infiltrative growth. See the muscle, how the muscle is getting pinched aside and just kind of being bored into by that malignant tumor. That's very common when you get a biopsy of a malignant tumor. When you have a CIS, that means carcinoma in situ. In situ, or Latin, it should be in situ, means in place. In place. Situ, that's where we get our word situation. The place where you find yourself. In situ, in place. So you have a pre-invasive tumor. It's got all the signs of being invasive, but it hasn't invaded yet. So you call that carcinoma in situ, and you usually want to have surgery as fast as you can to get rid of that. They're considered precancers. And they're usually considered high priority for extirpation. Surgical extirpation. Now, cancers, when we look at the cancer cell themselves, they have some hallmarks. here's some of those hallmarks. So first of all, they have evaded the mechanisms in the cell that are meant to suppress growth. So cells have two, roughly speaking, two groups of genes. One group of genes that promote growth, another group of genes that put the brakes on growth. The group of genes that promote growth are called proto-oncogenes. Other set of genes that cells have are called tumor suppressor genes. And they encode proteins that negatively regulate growth. So tumor cells have escaped the brakes put on growth by the tumor suppressor genes, usually because those tumor suppressor genes have loss of function mutations in them. And their proto-oncogenes have been transformed into much more active forms known as oncogenes. So the brake line has been cut and the accelerator is put down to the floor when it comes to growth. So this means you can take tumor cells and you can put them in culture in the laboratory and you can grow them indefinitely. In fact, we have a cell line called HeLa cells, H-E-L-L-A. HeLa stands for Helen Lux. Helen Lux was an African-American domestic worker who had cervical carcinoma, and scientists biopsied her cervical carcinoma, and without her approval or knowledge, shame on them, cultured it. And HeLa cell lines are now all over the world. They're one of the main cell lines worked on for cell culture. And the family actually turned around and they, after years, discovered what happened. They sued university, and they got some recompense. And there's an extremely good book by Rebecca McLeod called The Secret History of Helen Lux, that tells the story of the HeLa cell line and the story of this young lady who was wrong. And it's in many ways a very touching book. So they have this replicative immortality. And that's where we'll get for today. I'll pick this up on Monday, get it to where the podcast takes over, and then we'll go on to the next topic from there. All right, Emma, heal thyself.
All right, thank you
Tell Emma
again.
bye.
Thank you. Bye.
Okay. Be well.